Welcome back. So let's look at a few special steps that occur in meiosis 1. They're not going to be in meiosis 2, and they certainly were not part of mitosis. So I want to hone in on these, make sure you understand the details before we step back and go step by step through meiosis. So in the beginning, remember, an interphase. We had a chromosome from dad, and we had a chromosome from mom, and they replicated. And so now we have sister chromosomes from dad and sister chromatids from dad forming a chromosome, and sister chromatids from mom forming a chromosome. And we have 23 of those. So we've got 23 pairs or 46 chromosomes. Okay. So in prophase 1, what is going to happen is the sister chromatids number 1 from dad and sister chromatids number 1 from mom are going to get together, like for a meal. And number 2 from dad and number 2 from mom are going to get together. And that's going to happen for pairs 1 through 22, all the autosomes. It doesn't happen for the sex chromosomes, but it's going to happen for the first 22 chromosomes. Okay? And so when you look at it, you will see 1, 2, 3, 4 chromatids right next to each other, what we call a tetrad, because there are 4 of them. All right. So we've got these pairs of homologous chromosomes next to each other. They've got the centromere in the middle, and they got short arms at the top, and they've got their long arms at the bottom. And what you notice is they're kind of playing footsies under the table there. Yeah. Where nobody can see. Mom and dad with one of their legs, one of their long arms, is playing footsies under their table. And what happens is a break in the long arm of both a chromatid from dad and a chromatid from mom. And the genetic information from dad goes to mom, and the genetic information from mom goes to dad. So now one of dad's chromatids actually has some of mom's genes, and one of mom's chromatids actually has some of dad's genes. Okay. So this has allowed an exchange of genetic material between chromatids. So what's going to happen to the gametes after the results of this crossing over? Okay, so we started out with a tetrad with these homologous pairs. We did the crossing over. After meiosis 1, we're going to be separating dad's stuff from mom's stuff. We're going to be separating right down here in the middle. And then after meiosis 2, we're going to be separating the sister chromatids. So after meiosis 2, we end up with 4 gametes. 1, 2, 3, 4. So there'll be 1 chromatid per gamete cell. So when we look at it, 2 gametes are like the original. This first one all blue and the last one all purple are like the original. The all blue like an original from dad, the all purple like an original from mom. Whereas the middle 2 gametes are what we call recombinant. They have genes from both mom and dad. Yes, one of them is mostly dad with some mom, and the other one is mostly mom with some dad. But this is giving us genetic variability, which is important for evolution. So that was the first thing. The second thing happens in metaphase 1. Okay. So by metaphase 1, that has already happened because, as I said, this crossing over happened in prophase 1. So now we're moving to metaphase 1. And for simplicity, this slide is not showing the effects of crossing over. It's just showing regular chromosomes. And here, once again, we're showing red chromosomes and chromatids as if they're coming from mom, and blue as if they're coming from dad. And what happens here is called independent assortment or random alignment. In other words, this is not like kindergarten where you had to line up all the girls in one line and all the boys in one line. Here, what happens in the cell on the left is all the chromosomes from mom are on one side of the cell and the chromosomes from dad are on the other side of the cell. But on the second cell on the right, they're showing like, okay, so we've got a mom chromosome, the big chromosome number 1 from mom is on the left, but chromosome number 2 from mom is on the right, and vice versa. And so with only 2 chromosomes, there's really only 2 options. But remember, we've got 23 pairs of chromosomes. And so with 23 pairs of chromosomes, there are over 8 million possible combinations, because you could have 23 from mom on the left and 23 from dad on the right. You could have 22, the first 22 from mom on the left and then 1 from dad on the right. Or you can mix any one of those from mom. It doesn't have to be the last one from mom. It could be any one of those, or it could be 21 and 2, or 20 and 3, or 19 and 4, etc. And which ones they are can be totally random. So as a result, 2 gametes virtually never have the exact same combination. Because remember, there's 8.3 million possible combinations of these 23 chromosome pairs through this second thing known as random alignment or independent assortment. So let's look at the results of what happened with just 2 chromosomes. And once again, we're representing mom's chromosomes with red and dad's with blue. So with the first meiotic division, with meiosis 1, we are separating out the tetrads. And in this first one, all of dad's chromosomes are going to one cell and all of mom's are going to the second one. So in the second division, what we see is that 2 of the gametes end up with all of dad's genetic information and 2 of the gametes end up with all of mom's genetic information. With the same 2 only chromosomes, but splitting them so that one of dad's is on the left and one of dad's is on the right. After the meiosis 1, what we realize is chromosome number 1 is dad's and chromosome number 2 is mom. So after meiosis number 2, we have 2 cells that have chromosome 1 of dad and 2 cells that have chromosome number 1 of mom. And then the opposite would happen in the other 2 cells. Okay, so take a moment. This slide includes both crossing over and independent assortment. So it's going to take you a little bit longer to talk about both processes, but it's well worth your time to make sure you understand the processes, okay? And you've got a visual to help you with it. If you get confused, don't forget, make yourself a study group, meet with them, write to me in the Canvas inbox, ask for a Zoom meeting with me, go to the Learning Center, meet with the tutor and review this with the tutor. Make sure you understand this before you come back and go through the 8 steps of meiosis. So once you understand this, come on back and we'll go through all the steps of meiosis. Thank you once again for all your hard work in this class. I really appreciate it.